So good afternoon, everyone. Um, Today, I would like to talk about single molecular sensing with biological nanopore. Please don't hesitate to interrupt me during the talk if you have any questions. I think there are a few ways to do so. You can, in the reaction, you can raise your hands or leave your message directly in the chat. Okay, first I want to discuss the keywords in today's title. What is single molecular and what's the motivation behind? As we know, a molecular is the smallest unit of a substance that has all the properties. So to understand the world around us, how nature works, we need to study all these moleculars one by one. And second, I want to discuss what is nanopore. So if I need to use one short sentence to describe, basically it's a nanoscale channel, quite easy. And 1996, John Kasanovich used alpha aerolysin, which is this pore structure showed here, proved the concept that for detection the single-strand DNA, we can use this kind of protein to do so. Since then, many proteins from nature has been explored and utilized as biological nanopore sensor for different applications. In addition to such nature material, some solid state, synthetic, even the origami has been used to fabricate a suitable nanochannel structures. How does it works? How nanopore detect the target molecular? Here I use classic alpha aerolysin as an example. First, for such biological nanopore system, you need to have a lipid bilayer to support or hold the nanopore, which is insulated. And then the whole system is in a chamber, which is filled with a high salt concentration solution. Normally it's 1 molar KCl. The green dot here is chloride anion, and the yellow is the potassium cation. After we apply a voltage, you can imagine the ions will flow through the channel because here is the only way to do. And this will lead to a constant open pore current. If we have a single strand DNA to transport through the channel, you can imagine when it stays inside the channel, it will take the space of the ions which is supposed to be there. So we got dramatically decrease the current. When it leaves the channel, the current will go back. So if we have the other protein or molecular pass through, we will have the other current signals. Depending different amplitude, dwell time, or shape of these signals, we can understand the properties of the target moleculars. And for this method, it's very good for the complicated system because you can imagine you can mix as many target molecular you're interested to let them go through because you're looking for the current signatures. This has been developed for efficient reading long-strand DNA. When a single-strand DNA transports through the pore, different nucleobases block the current at different levels. Therefore, the order of ATCG can be read in real time without the need of the PCR and additional base labeling. More importantly, this technique is electric readout, which means cheap and easy to be massively parallelized. In principle, there is no limit for the reading length, which is a very unique feature for this technology. And also can be used for polymer or protein because what you are reading is the difference of the ionic current, which induced by target molecular. So no limitation for molecular you are interested to detect. And the last and most important point is that this approach is conducted under the solution. So although it's high salt concentration, but compared to the other method, it's more close to the native environment of the biomoleculars. In my lab, we focus on engineer and design new biological nanopores, and we have three main focus of application, including decoding digital polymers or protein biomarker detection. And as a nanopore group, of course, we're interested in DNA or protein sequence as long-term interest. For engineered biological nanopore, today I will talk about only engineered nanopore and the digital polymer, these two parts. For engineered biological nanopore, we did it based on the aerolysin protein, a founding member of the beta pore forming toxin, which structure has been well studied in past 30 years. Actually, most experts are located in Switzerland, EPFL, and the Bern University. As a nanopore sensor, aerolysin has two fundamental advantages I want to highlight here. First, high sensitivity. Compared to the other mostly used or commercialized nanopore, its diameter is the narrowest. Therefore, it provides a high sensitivity. My previous work and work from the colleagues have proved that it can directly identify single nuclear base variation, different lengths of the peptides, or just different types of amino acid, which is quite impressive. More importantly, this heptamer structure is super robust. It is resistant to the high temperature or denatured condition, or even very harsh pH conditions. When we purify this protein, we saw the heptamer structure in the SDA's page directly. So I will show that part of data later, so you will know how robust this pore is. And very recently, our study realized that the nice feature of the aerolysin nanopore actually are determined by the 4 very constriction point. I grouped them into 2 regions here, top and bottom. In this field, as what has been done in the alpha hemolysin and MSPA, mutation is a very powerful genetic approach to engineer the amino acid precisely and thus have a tuned property based on the need of the sensing. We rationally engineered amino acid of aerolysin and 4 important points with consideration of steric hidance and electrostatic, which are the 2 most important factors for nanopore sensing. As you can see here, we mutated them to the alanine to make it wider, or we mutated to the tryptophan to try to make it narrower. If you notice, all of them are positively charged amino acid, so we mutate them to Q or N to make them neutral, or change to the negative amino acid E, or go back to different positive amino acid to see the difference for molecular sensing. Instead of purify all these proteins, we used the in silico method first, molecular dynamic simulation, to study them. As you can see here, first we evaluate the pore structure variation. If we mutate this position to alanine, the diameter will change from 12 angstrom to 70 angstrom, almost. And for 220 position, it changed slightly bigger from 7 to 17 to 16 angstrom. Anyway, you will see the alanine will really make the pore wider. We did for all of the mutation we proposed, and we found for tryptophan, it's not as what we expected. So this is the red dot you see here. The black is the Y type, so almost the diameter doesn't change or slightly bigger. So it doesn't make the pore narrower, almost no change. So we go back to simulation because the tryptophan is hydrophobic, so the residue like to go close to the wall rather than extend to the channel. So that's why we got even wider pore rather than narrow pore. So it's not always works like what we want. And the other very interesting phenomena we observed is here. So I list the mutation on the bottom, y-axis. You can see the mutation at the bottom, K238A, make the diameter of the top region, which is red dot here, 220 region, narrower than before. So it basically, you mutated bottom, but the top become narrower. And more interestingly, we did the RMSF calculation of this different position. The mutation at the bottom actually make the fluctuation of the 220 region more rigid. So you make it narrow and rigid, which is perfect for molecular sensing. And now we calculate the ionic current conductance, which exactly as what we did with the experiment, open pore current, but of course in a short time scale, which is nanosecond. And the noise is high, so we need to smooth it to see the current trace. So you can see this is around 100 picoA. You have different way to compute this current. You can do histogram or slope fitting of the accumulate current. Anyway, you will get the same number. We did it for all of the mutation we're interested. So I want to highlight this guy here, 220A. If you remember, we make the pore super wide with this mutation. However, we didn't get more open pore current. How it possible? It's wider, but less ions go through. So we got confused, and we want to validate our AMD simulation result first, if it's exactly right or wrong. So we select a few of the mutation, express them, and purify them. And here is what I mentioned before. You see, this is SDS page, and we see the haptomer structure very well, which means super robust. And we did very standard method as nanopore measurement. We purify the monomer protein, will have a pre-structure first, and then it will insert into the lipid bilayer, have the pore structure. And here is the open pore current I collect from them. This is comparison from the experimental result and MD. You can see more or less we have a good tendency. It's comparable. And we do have very low current with 220A. So if you do the IV curve, this is a result from the experiment. So you see all of mutation at the top region actually make the current much lower than the other side. And the mutation at the bottom seems then change that obvious as here. So here, there is a good point of MD because we can separate current of the chloride and potassium separately to evaluate what happened there. So at the positive voltage, you see clearly all of current dominated by the chloride current rather than potassium. If you go, so for 220A, which we're interested because it loses a lot of chloride current. Although it increases slightly for the potassium current, but this chloride current is dominates the total current here. So I would say because the arginine to alanine, so the mouth of the pore lose the positive charge, so it loses the attraction of the chloride current. That's why we got wider pore but low current. If you go negative voltage, we do observe for the top mutation, it becomes to the potassium dominated current. So that's why it has wider pore at the negative potential. And the mutation at the bottom region doesn't change that much. So I would say the current selectivity seems controlled by the top region of the pore electrostatic. We also can do eye density map to see which position it really makes sense. So you see here for the top mutation, if you start from Y type to Q neutral and to E, you really lose the chloride current here. That's why we lose the open pore current. So after all of this understanding, what we want to do is molecular sensing. So we go to the DNA detection directly to see what happened there. You can see compared to the Y type, for the top mutation pore, we lose good signal to noise ratio. The signal becomes super short and not even uniform. if we do dwell time versus applied voltage, they almost doesn't change. So in this field, it's supposed to be something just bumping on the top of the pore and just go away. It's not translocation even. And for all of the mutation of the bottom region, we get very good signal. For some of them, the dwell time even longer than Y type, because longer dwell time means you can estimate the current level much more precisely. So it's always a good feature in nanopore studies. If we have a look for dwell time versus applied voltage, you see they decrease. If you have an increased voltage, which makes sense, it probably is the translocation. If you give more force, it translocates much faster. There is one exceptional guy is K238Q before 160 milliwatt. If you give more force, driving force, it will stuck in the pore much longer. At some point, it start to translocate. So there must be some specific interaction there, which we want to dig more in the future to understand. So we want to understand this DNA capture problem. Can we learn from the chloride ions capture? So if it's related to the electrostatic of the top region or not. Here we plot the electrostatic potential. Top is the minus 100 milliwatt, and here is 0. Instead of a linear change, we do see the electric field has been changed by the pore structure and the pore electrostatic. So here we see obviously the mutation at the top. Why it cannot capture DNA? Because instead of a driving force, it gives a opposite direction electric field. So it basically repel all of your molecules. That's why we didn't get a signal. For the dwell time, we want to understand which part it is related to. So we compared the dwell time and the diameter and two region, top region and bottom region. You can see all of the mutation happened bottom region, but we do observe a good correlation at the top diameter, which also makes sense with what we figured out before, because you make the top region more narrow. So it determined the narrowest part of the pore rather than where you did the mutation. As I say, this technique is not, how to say, specific to your target molecular. So it should work for the same for the other molecular. So we tried the peptide here, it's negative charged. We observed the same correlation as we observed from the DNA. And if you reverse to have some negatively charged peptide, of course, before the top mutation cannot capture DNA, but now it become positive peptide, it should have a very good signal to noise ratio here. And this is actually what we collect here. The top mutation now can used for this positive charged peptide sensing. If you change the voltage, the dwell time becomes shorter, so it also proved to be a translocation. The diameter and the dwell time also fit very well with the 220 region. If I do a quick conclusion here, we understand the sensing ability of the aerolysin is mostly controlled by the narrow diameter of the top beta barrier, which is 220. And the selectivity of the pore are dominated by the electrostatic of the top region. The whole region play a role in these selectivities. If no question, we move to application, what we did with this engineered nanopores. As we well know, the digital data continuously accumulate and in our society, so higher density storage are necessary. Although the research on semiconductors has been dramatically improved in past 20 years, this technology cannot meet the exponential growth of the digital data production and storage. Meantime, it breeds the other issue, which is energy cost. This is a report from 3 years ago. At that time, the electricity used to maintain one data storage center in UK is already more than the national energy consumption of some countries. And this demand will increase 20 times in next 10 years. So for scientists, the new strategy of coding and decoding have to be explored. One of them is to encode the information into the molecular, like what nature did for the genetic information storage, DNA. This kind of biological inspired platform has two fundamental advantages. One is long-term storage. Unlike the magnetic tapes, it can last for a few hundred or thousand years, and higher information density. Compared to the hard disk, DNA data storage density is million times higher. If we use some smaller chemical subunit, it can be pushed 100 million times higher. As you can see here is a work from George Church. They used CRISPR-Cas system to encode a GIF into the bacteria and not decoded by the DNA sequence approaches. So for molecular data storage, I think DNA is an obvious starting choice. However, there is little space for optimization and innovation of DNA structure because all the ATCG. Alternatively, precise synthesis of sequence-defined polymer has recently opened up the possibility to two very important parameters like storage capacity, density, readability, and writing speed. But for synthetic polymers, so far it only be read by the mass spectrum, which is a huge machine. So it's impossible to do the real-time data storage reading and writing in the future. And a few years ago, our collaborator from the CNRS came to us and say, why not try the nanopore to sequence these polymers that carry in the digital information? That's how we start. So first we go back to our engineered aerolysin library. We choose the one we think had the best resolution for molecular sensing in general because there's no specificity, so should be work for polymer as well. And here I want to say, because both aerolysin nanopore part and the polymer part can be tailored, can be designed in a lab. So it gives us a lot of possibility to optimize the structure from both sides. Here, we use the phosphate backbone because it gives you negative charge, so it can be driven by electric field. And we also optimize the bit one for few structures because we want it to be bulky, so can be separated by the bit 0. Here the two we choose in the end. If we add only bit 0 into the nanopore system, barely get signals. If we add the bit 1, we get some signal, but signal to noise ratio is not good to do the real decoding. So we go back to our previous study because we know very well in aerolysin system, the A4 have a very good signal readout in this pore. So we add the deoxyandrogen at the terminal of our polymers to see if it can increase the resolution or not. This additional nucleotide creates highly detectable current blockage signal because before 0 we cannot detect, but now it's blocked half of the pore and very obvious signal. If we have a bit 1 was inserted to the polymer stand, so we have much lower blockage current now and a very clear 5-level signal observed here. To understand the relationship between the polymer chemical structure here and the current level we observed. So we collect more than 10,000 events for statistical analysis. The relative current fitted by the Gauss distribution and show 5 distinct levels of the value, while the dwell time fitted by the exponential function. The relative current value of the level 2 and 4 is more or less the same and similar as the bit 0. So we think they caused by the bit 0. And level 1, 3, and 5 are quite similar. And the dwell time, yes, the dwell time is more or less the same. So according to our previous study, there is a very strong correlation between the physical size of translocated molecular and the relative current value. So we think this very low current blockage from beginning and end should comes from the nuclear base, which is much larger than our chemicals in middle. And the level 3 is from the BioK bit 1. Here, I just want to show you, here is the clustering of the signal from this polymer. So I just want to show you, because it's single molecule behavior, so it can be super dynamic. It can be super fast at the beginning or super slow at the beginning. All can be happen. That's why we often do the statistical analysis to double confirm what we observed is happened many, many times in the whole system. So next, to optimize the polymer design and further understanding the influence of the terminal nuclear base for the decoding, a series of polymers has which replace the terminals with different nuclear base and different position, G and T has been started. So if we compare the polymers from the left panel, you can see if you replace the CC at the 5 end, you have less current at the end. If you place it at the 3 side, you have less current at the 3 side. If you replace all of them by C, you basically lose the current at the beginning and the end. So it further confirms the current from the beginning and the end comes from the nuclear base. So with this, we think this strategy not only can read the bit 1 and 0 in the middle, it also can read the nucleotide and the terminals and also the orders. To systematically evaluate the difference of this nuclear base for the reading of the polymer, we calculate the mean dwell time, because longer dwell time allow a more accurate determination of the current level. So the longer the better. This one is the best from here. And we also have a look for the mean current variation. So higher variation promote a higher read accuracy for each bit. So in the end, combine these two parameters, we choose the AA in the end, which is we use it in the following design. And we then test the sensitivity of the pore for detecting bit 1 monomer when it spans the 5 available positions along the whole backbone. So for this task, we developed a deep learning approach to process the data, current signals, and the assignment of the polymers to automatically classify a much larger fraction of the events with a high accuracy. So this result show us that the detection of the bit 1 is difficult when it frankly directed to the terminal nuclear base. If we only use the innermost positions, then the accuracy can be as high as 97%. So we only use the inner position for the following reading single bit. Instead of only 3 bits, we also consider all of 1 bit, 2 bit, and 4 bits length polymers. And we tried all of combination for this different lens. And here is deep learning result. We do observe a very accuracy decrease for a longer polymers, but the accuracy we're talking here always related to selection percentage, how many data you use to predict your result. I would say it's okay because 90% selection, you have higher than 90% accuracy. And we also, to validate this deep learning platform, we also did the blind test and mixture sample test. So more or less, we proved the method itself is good enough to assign all of these polymers. And here is a statistical analysis. And to give better landscape, I also include few longer polymers like 5 or 6 beads. So in general, we observe a decrease dwell time with longer lengths. It also makes sense because the longer polymer it is, the more negative charge it carries. So it will translocate the pore much faster. But we observe this here for the 4 beads polymer, although it's become faster, but the current variation is still higher compared to the 2 beads length and the 3 beads length. So it means we do have more information there, but like for the longer polymer, we don't know. We are basically balance the speed and resolution. So we are still working on this to how to reduce the negative charge density in the backbone to make it more readable for our nanopore system. In the meantime, we also try to mutate our aerolysin nanopore to make it much narrower or more sensitive to try to make the reading easier. So both parts are still working together to get better result. And for this, I hope I convinced you that this bio-inspired writing and the reading platform we have developed can be a solution for data storage. And of course, it's not limited to data storage. It also can be used before we really reach there. It also can be used for cybersecurity or anti-quarter fitting system, which based on moleculars. As I mentioned, we have the other research interest, which is an ongoing project, collaborate with Hilala Shu in EPFL. We are detecting the protein biomarkers. And for this project, we hope we can transfer it to the clinical trial to do some diagnosis to really make it useful. And for DNA and protein sequence, we also have ongoing project working, trying to push, because nanopore DNA sequence method so far haven't taken the whole market because the accuracy. The accuracy is not as high as it should be. We don't have single level for each nuclear base. We have combination, like 4 bases will influence the current blockage. So we're trying to really push to get one current level correspond to one nuclear base. And for protein sequence, this field just starts. So there are so many questions need to be addressed in this field. And currently we have 2 PhD open position in our lab, which affiliated to the doctor program in biology, biotechnology, and bioengineer. So anyone, if you're interested in this topic, please contact me and we can chat more on this. In the end, I would like to thank all of my colleagues, collaborators, my former boss, Madeo Doppler. He gave me all of opportunity to carry on all of this project and also support me now in my project, Prima project. And Professor Jean-François Louty basically provide all of digital polymers for us. And these polymers have been produced by Nicolas and Aziz. Professor Alexander Randovich gave me general support for all of this all of nanopore topic because she is also a specialist on the solid state nanopore. And Nulia we're working together on the AMD simulation part. And also fundings, of course. And I think my talk is done. Any question, I'm happy to take.